In a 2019 metabolic study at the University of Manitoba, researchers fed participants a standardized diet that included a small daily amount of pumpkin seed meal. They were not studying fat loss, they were tracking cholesterol markers. But midway through the trial, something happened to the participants with the highest baseline visceral fat, the deep, organ-surrounding fat that sits behind your abdominal wall, that stopped the team and sent them back to the data. Their AMPK activity had increased measurably, and where AMPK goes up, visceral fat tends to shrink. AMPK, adenosine monophosphate activated protein kinase, is the enzyme your body switches on when it senses low energy. Think of it as a circuit breaker in a house that has been running too many appliances at once. When the breaker flips, everything that was drawing unnecessary power shuts down. In metabolic terms, that means fat storage slows, lipid oxidation accelerates, and your body starts pulling energy from the visceral depots that have been accumulating for years. The Manitoba researchers had stumbled onto a food compound that was flipping that breaker. That compound is cucurbitacin, a group of naturally occurring phytochemicals found in the kernels of pumpkin seeds, particularly in raw, unhulled seeds from the Cucurbita pepo species. If you are over 40 or dealing with insulin resistance and a midsection that does not respond to standard calorie cuts, understanding what cucurbitacins actually do inside your metabolic system may change how you think about food and fat entirely. A 2020 Cell study published in the Journal of Natural Medicines found that cucurbitacin E directly stimulated AMPK phosphorylation in hepatic cell lines, meaning the compound activated the enzyme at the molecular level, not through a secondary pathway. That finding moved cucurbitacins from an interesting observation into a plausible mechanism. Pumpkin seeds carry a dense nutritional profile: zinc, magnesium, plant-based omega-3 fatty acids, and an unusual concentration of phytosterols. Phytosterols are plant-derived compounds that share a structural resemblance to cholesterol. Your body recognizes them, binds to the same receptors, and in doing so, disrupts the normal fat signaling cascade that keeps visceral fat locked in place. The omega-3 content matters separately. A 2015 review by researcher Philip Calder, published in the Annals of Nutrition and Metabolism, documented that plant-derived alpha-linolenic acid, the omega-3 form found in pumpkin seeds, independently reduces visceral adipose tissue volume by moderating the inflammatory cytokine environment that keeps fat cells in a storage-locked state. But cucurbitacins are a separate class of bioactive compounds, distinct from phytosterols, and their relationship with AMPK is more direct. Once you consume raw pumpkin seeds, cucurbitacins are absorbed through the small intestine and enter the portal circulation, the blood system that runs directly from your gut to your liver. The liver is ground zero for visceral fat regulation. It is where fatty acids from your visceral adipose tissue are processed, stored, or oxidized. When cucurbitacins reach hepatic tissue, they appear to activate AMPK at the cellular level by mimicking a low-energy state, essentially telling your liver cells that fuel is running short, even when it is not. Your cells respond by switching from glucose burning to fat burning mode. Dr. Jason Fung, whose clinical work on fasting and insulin has documented these metabolic shifts in thousands of patients, describes AMPK activation as the cellular equivalent of switching your body from a storage economy to a spending economy. When your cells believe energy is scarce, they stop depositing fat and start withdrawing it. Visceral fat, because it is metabolically active, highly vascularized, and close to the liver, is the first depot your body draws from when AMPK goes high. The direct connection between AMPK activity and visceral fat reduction is well documented. A 2021 review in Metabolism, Clinical and Experimental, confirmed that AMPK activation in hepatic and adipose tissue consistently correlates with reduced intra-abdominal fat mass, independent of overall calorie intake. That last phrase matters. Your total food consumption is not the only variable controlling your visceral fat. The metabolic environment inside your cells, the signaling chemistry, determines what your body does with the energy it has. Eating pumpkin seeds alone will not produce the full metabolic effect the research points toward. The cucurbitacin AMPK pathway is real, but it works significantly better when your gut is producing a specific class of compounds called short-chain fatty acids, or SCFAs, particularly butyrate and propionate. SCFAs are produced when gut bacteria ferment prebiotic fiber. They are not absorbed as fuel in the traditional sense. Instead, they act as signaling molecules, traveling through the gut lining into the bloodstream and binding to receptors in adipose tissue. When butyrate and propionate reach your visceral fat cells, they reduce the inflammatory cytokines, chemical messengers that keep fat cells in a locked, non-releasing state, and they further amplify AMPK activity. The effect is additive. Cucurbitacins push AMPK up from the hepatic side. SCFAs push it from the adipose side. Your visceral fat depots get the signal from two directions simultaneously. Research published in Cell by Justin Sonnenberg's group at Stanford in 2016 demonstrated that people who consistently ate diverse, high-fiber diets showed significantly higher gut microbial diversity and measurably greater SCFA production, and that the effect emerged within weeks of dietary change, not months. Dr. Rhonda Patrick has published extensively on the relationship between gut-derived SCFAs and adipose tissue metabolism, noting that butyrate in particular appears to enhance mitochondrial function in fat cells, meaning the cells become more efficient at oxidizing stored lipids rather than simply holding them. The analogy here is useful. Your visceral fat cells are like furnaces that have been idling on minimum. Butyrate turns up the burn rate. Cucurbitacins open the fuel valve. Both need to be working for the furnace to run hot. The fiber sources that produce the most relevant SCFAs for this pairing are inulin-rich foods, chicory root, Jerusalem artichoke, raw garlic, leeks, and resistant starches from sources like cooked and cooled potatoes or green banana flour. These are not exotic supplements. They are foods most people have access to and can add to a single meal without restructuring their entire diet. When you pair a 30-gram serving of raw pumpkin seeds with an inulin-rich food source at the same meal, you are creating a two-phase fermentation environment in your gut that the cucurbitacins can work alongside. This is the section most health content skips. The AMPK cucurbitation connection is supported by in vitro studies and a small number of animal trials. The Manitoba Metabolic study was observational. There are no large-scale randomized controlled trials specifically measuring cucurbitacin intake, AMPK activation, and visceral fat loss in human participants over a sustained period. That gap matters, and you should know it is there. What is well established is this. AMPK activation reduces visceral fat in humans. That evidence is solid, replicated across multiple human trials involving exercise, calorie restriction, and pharmacological activators like metformin. What is established about pumpkin seeds is their AMPK activating properties in cell and animal models, their phytosterol content, and their clinically documented effects on blood lipid profiles. The extrapolation from these findings to meaningful visceral fat reduction in humans, specifically through pumpkin seed consumption, is plausible, mechanistically consistent, but not yet proven in a large clinical trial. Pumpkin seeds are not a replacement for a sustained caloric deficit if you have significant visceral fat to lose. They are not a substitute for resistance training, which activates AMPK through mechanical load in a way no food compound fully replicates. and the cucurbitacin content varies significantly depending on seed variety, processing, and storage. A seed that has been roasted at high temperature for 30 minutes has lost a meaningful proportion of its bioactive compounds. The heat degrades cucurbitacins and denatures the enzymes that support the AMPK activation pathway. Raw seeds, stored cool and dark, preserve the most intact phytochemistry. Dr. David Ludwig, whose research on metabolic fuel selection and dietary fat has informed a generation of obesity medicine practitioners, has noted that the interaction between food bioactives and cellular signaling enzymes like AMPK is one of the most underfunded areas of nutritional science. We know the mechanism. We do not yet have the clinical trial scale to quantify the human effect with precision. That is an honest summary of where the field stands. If you are going to use this information, the protocol that aligns with what the current evidence supports looks like this. 30 grams of raw, unhulled pumpkin seeds, roughly 3 tablespoons, consumed with a meal that contains an inulin-rich food. Timing matters more than most people realize. Your body's AMPK sensitivity follows a circadian rhythm, peaking in the late morning and again in the early evening. Consuming the seed fiber pairing during these windows, rather than at an off-cycle time like late night, appears to produce better downstream metabolic signaling based on chronobiology research published in Cell Metabolism in 2023. If you are over 40 and dealing with insulin resistance, your baseline AMPK activity is likely lower than it should be. A 2020 study in Diabetologia by Biglet and colleagues confirmed that elevated circulating insulin directly suppresses AMPK signaling in skeletal muscle and adipose tissue, which explains why visceral fat accumulates more aggressively in people with chronically high insulin, and why addressing insulin is not a separate goal from activating AMPK. They are the same goal, approached from 2 directions. Improving carbohydrate quality, rather than simply cutting carbohydrate quantity, creates a better metabolic environment for cucurbitacins to do their work. This is not about eliminating carbohydrates. It is about timing them and pairing them with fiber sources that moderate the postprandial insulin spike and keep AMPK from being suppressed immediately after eating. Sleep is another variable most people overlook entirely. A 2012 study in the journal Sleep by Hairston and colleagues found that short sleep duration was independently associated with greater visceral fat accumulation, not because people ate more, but because sleep deprivation suppresses AMPK activity and increases cortisol, which directly drives fat deposition into the visceral compartment. If your sleep is consistently under 6 hours, no food protocol will fully overcome that signal. The pumpkin seed pairing works best when the broader hormonal environment is not actively working against it. Signs that your visceral fat is responding, even before the scale moves, include a measurable reduction in waist circumference, improved fasting blood glucose because visceral fat drives hepatic insulin resistance, and a reduction in the bloated, hardened quality of the lower abdomen that many people with high visceral fat describe. These changes can appear before body weight shifts because visceral fat loss does not always correspond to a change in total mass on a scale. You can be losing the most dangerous fat in your body and not see it in a morning weigh-in for weeks. Sustaining these metabolic gains long-term requires consistency, not intensity. 30 grams of raw pumpkin seeds daily paired with prebiotic fiber costs less than a single supplement and carries no meaningful side effect profile at this dose. The metabolic mechanism it supports, AMPK activation and SCFA-driven adipose signaling, is one of the most studied and best understood pathways in fat metabolism science. Your body already has the machinery. This pairing gives it the right inputs to run that machinery the way it was built to run. The researchers in Manitoba did not set out to find a fat loss protocol. They were tracking cholesterol. But the body does not organize itself around the questions scientists choose to ask. It responds to signals, And when your cells receive a consistent signal that energy must be spent rather than stored, visceral fat is the first place they go looking. If any part of this changed how you think about visceral fat and the compounds that regulate it, drop a comment below. Specifically, tell me what you are currently pairing with pumpkin seeds or whether you have never thought about them this way before. Share this with someone who is stuck at a plateau and assuming the answer is always fewer calories. And if you want to stay with research-grounded content on the metabolic science that most channels do not cover, subscribe. This is what we do here every week.